Salam Sejahtera to everyone. I'm Vanessa Midfulton, and today I will present my final year proposal with the title An Assessment of Cholera as Microbial Barometer of Climate Change in Malaysia, supervised by Associate Professor Dr. Leslie Morris Dino. First, I will explain briefly on the introduction. Cholera is known as diarrheal disease that affects the gastrointestinal of the host. It is caused by the bacterium Vibrio cholera. So this bacteria produces toxins that leads to various symptoms such as vomit, diarrhea, fluid loss, and electrolyte reduction. Cholera is mostly happen because of poor sanitary conditions, inadequate access to safe water, consumption of contaminated food, and our main concern is changes in environmental factors caused by climate change. According to Hassan et al., rainfall, temperature, and humidity does impact cholera spread in Malaysia, but for each state are varied in what causes cholera to arise due to various climate factors. Most of the cases arises in Malaysia are occurring near the coastal areas. Next is the objectives. So here we focus on three objectives. First is to determine the association of climate change that affects cholera incidence by conducting statistical analysis, that is, Poisson generalized linear model, multicollinearity tests, and also sensitivity testing. Secondly is to analyze the effectiveness of cholera as a microbial barometer of climate change by analyzing the data of cholera cases with meteorological data from 2004 to 2014. And thirdly is to predict the future implications of cholera by evaluating the data involved. So next, we will go to the literature review. So we will take a look on the discovery of cholera first. In 1854, Passini and Snow are both interested to find what causative agent that causing cholera to occur. Passini had observed dead autopsy records and samples from cholera patients, so he analyzed millions of elements which he called as vibrants. Meanwhile, Snow investigated water samples from wells and he verified the presence of white flocculent particles by using a microscope. Snow believed that cholera occurs via food and water contamination, but he could not verify his hypothesis. So in 1854, another discovery was made by Robert Koch, who performed several postmortems and discovered a bacillus in the intestinal mucosa that was exclusively found in the bodies of cholera victims. He isolated the bacillus in pure culture and it has a comma-shaped look. So this bacillus could grow in moist and damp soils and is sensitive to drying and mild acid solutions. For the history of cholera, so far cholera has caused 7 pandemics around the world and has greatly impacted on many nations. Now I will explain briefly on the mechanism of cholera toxin. Cholera toxin is made up of 6 protein subunits, which consists of 1A subunit and 5B subunit. So these subunits are connected by the sulfide bond. So for the B subunit, when it is produced, it binds to monocelluloganglioside GM1, which causes fluid loss by chem-mediated anion secretion. It also causes suppression of electroneutral sodium chloride uptake. According to Ramamurthy et al., CT gene, called as CTX6, found in the CT cascade, a prophage that has been demonstrated to integrate into the Vibrio cholera chromosome via a receptor called toxin co-regulated pili. So this cholera toxin responsible in causing some symptoms that are experienced by the cholera patients. Next, we will take a look on mechanism of heat-stable enterotoxin, or called as ST. So this heat-stable enterotoxin plays a role by sending signals to the cyclic guanosine monophosphate CGMP pathways, which could trigger cystic fibrosis transmembrane conductance regulator, or often called as CFTR chloride channel. So ST is also important in elevating the intracellular CGMP, which could lead to an increase in anion discharge and causing diarrhea in cholera patients. But according to Ramamurthy et al., it is extremely unusual for Vibrio cholera 01 strains to express heat-stable enterotoxin. Next, I will explain briefly on the epidemiology of cholera. Cholera is endemic in many areas of Asia and Africa. Besides, changes in the environment have a crucial role in cholera epidemiology. For example, alteration in surface water temperature and terrestrial nutrient flow could influence phytoplankton and zooplankton to increase, resulting in a rise in Vibrio cholera. In human patients, Infectious dosage of Vibrio cholera of 1 was determined to be 10 to the power of 5 to 2 to 10 to the power of 8, although it might be as low as 10 to the power of 3 when eclodia is present. In humans, infectious dosage of Vibrio cholera varies significantly, relying on the bacterial strain and type of the host. Next, I will explain on climate change. According to UNFCCC, climate changes are caused by human activities, whether directly or indirectly affecting our atmospheric compositions. According to Haliza et al., climate change can be defined as unobservable changes that occur in the condition of our climate, which continues throughout a lengthy period, generally for decades or longer. For the next point, we will take a look on the environmental factors that influence cholera spread. So we will take a look on 4 factors that influence cholera growth. First is increased temperature. It can speed up the pathogen growth and multiplication in their natural environments, especially in raw food like shellfish, water, and the environment. Secondly is heavy rainfall. Worse water contamination with raw and treated water sources, which can cause floods and impact water quality and sewage systems. Thirdly is rainfall. As climate change rises, low amount of rain can lead to drought, which is one of the factors for Vibrio cholera to spread more. This contribute to cholera incidence. And lastly is decrease in pH level. pH level of ocean are reduced when huge amount of carbon dioxide are absorbed. This lead to ocean acidification and increase the Vibrio cholera replication. Next, I will explain briefly on the effects of climate change on cholera infection in Malaysia. Hassan et al. Stated that cholera infections are most likely to happen near the coastal areas. For example, like in Sabah, Sabah has higher rate of cholera infection cases, and most are coming from near the coastal areas. If compared to the other states, had fewer cases than Sabah. So there are 2 factors that leads to cholera infection in Malaysia. First is the ocean temperatures. This is a significant factor that lead to cholera cases, for example, like in Sabah, because rises in temperature can cause planktonic species to bloom, including blue-green algae, diatoms, and many more. So the shellfish will feed on these planktonic species and the algae as the food source, so it makes them to become curious that infected with Vibrio cholera. For the second factor is heavy rainfall. Some states in West Peninsular Malaysia are prone to flooding, for example, like in Kelantan and Terengganu. Meanwhile, other peninsular states have very rare cases of cholera occur. Next is the weather patterns in Malaysia. Malaysia is a tropical country with an average annual rainfall that is between 2,000 to 200 to 2,500 millimeters, and an average temperature that is between 21 degrees Celsius to 32 degrees Celsius. There are generally 2 types of monsoons that always occurred in Malaysia, that are southwest and northeast monsoon. Southwest monsoon are likely to have a higher temperature if compared to the northeast monsoon, which has a little lesser temperatures. According to Hassan et al., some states on West Malaysia's east coast, including Pahang, Kelantan, and Terengganu, experience more precipitation during the northeast monsoon, and that is why these states are very prone to flood. Next is the methodology. The study area is basically involving all the states in Malaysia, and for the cholera cases, will be analyzed by examining the data taken from 2004 to 2014. Data collection is very important for both cholera cases and meteorological data because we want to know what are the relationship between these two. Next is the statistical analysis. In Malaysia, we have 39 stations for temperature and humidity and 115 precipitation stations. So these stations basically responsible to record the data of these factors. For the missing data, for this study, it will be categorized as missing completely at random. This study also will use Poisson generalized linear model, which helps us to determine the association between the monthly lag 0 to lag 2 month for the meteorological data and the incidence of cholera cases. Time stratified model and Fourier terms will also use to regulate long-term trends and seasonality. And IBM SPSS statistics version 22 will be used to conduct regression analysis. Here we can see that the formula of Poisson generalized linear model which will be used. Next is the multicollinearity test. Multicollinearity test is to determine between the multicollinearity existence between the analyzed climatic variables. So there are several measures that we need to take a note of. First is variance inflation factor, second corrected inflection factor, and tolerance. And lastly is sensitivity testing. Sensitivity testing is to determine the sensitivity of the important parameters by substituting one of the variables that are used in the study, and like data will be used. For the expected outcomes, it is expected that we're able to relate the effect of climate change with the incidence of cholera in Malaysia, as well as to determine Vibrio cholera as an effective microbial barometer of climate change. Using Poisson generalized linear model helps us to determine the association between monthly leg 0 to leg 2 month meteorological data and the incidence cases of cholera in Malaysia. On the other hand, multicollinearity tests and sensitivity testing also enable us to find out whether Vibrio cholera is a good microbial barometer of climate change. In this study, the research from statistical analysis and multicollinearity tests can be useful to researchers to enrich the information of cholera relationship with climate change. Thus, it also enables the free extension of data to the scientific community in the future. The results from the sensitivity testing justify the validity of changes in environmental factors as causes of cholera cases. This is important in facilitating the studies on the role of climate change in cholera incidence. Hence, it can develop ways to mitigate cholera cases in the future. Next is the work schedule. This schedule shows the proposed timeline of the work associated with the proposed study, starting from October 2021 and end in June 2022. Lastly is the references. So all of the journal articles and internet sources that are used are listed here. This is the end of my presentation. Thank you for watching.